So the final paper in this session is presented by Jonathan Hardman. Human scalp hair follicles engage in and depend on autophagy for hair growth and maintenance.
Thank you for having me. This talk's actually a prequel to one that occurred on Wednesday, but hopefully I'm going to try and convince you all that human hair follicles use autophagy to maintain themselves in anagen for long periods of time. So how is, one of the big questions in hair follicle biology is how is the follicle able to remain growing for such a long period of time, particularly in human hair follicles? And this is really important because by understanding how these basic mechanisms, we can find new areas to target to try and treat diseases such as androgenetic alopecia, telogen fluvium, and alopecia areata, which are hair cycle disorders. Well, what is autophagy? Well, autophagy is a cellular recycling process. It's particularly common in cells that are very, very stressed or undergoing cellular differentiation, and it's very important for just general cell homeostasis. Now, during the autophagy process, we get an isolation membrane here, which engulfs old proteins and organelles that are no longer working, and these are surrounded and by a membrane becoming the autophagosome. Now, this fuses with a lysosome and is targeted for degradation, and the cycle keeps repeating over and over again. This is often in tissues that have a lot of stress or a lot of reaction species production. And just to point out here as well, some of the key proteins involved in this process are LC3 and ATG5, which are important and found in the membranes of the autophagosomes. And why would we think this was even occurring? Well, anagen is a highly energy-using and protein-consuming process, and there's also high levels of reactive oxygen species, which suggests there may be a link. Moreover, chloroquine has been known to cause hair loss, and this is also a very, very powerful autophagy inhibitor. So this gave us our first idea that we may be able to link the two processes. So we went into this experiments with the hypothesis that human hair follicles do use autophagy, and this helps them live for a long time in anagen. And we wanted to find out as well, if it is true, can we manipulate this and influence the hair cycle? So our first experimental question was, do follicles even have the machinery necessary for this process? And to do this, we used human occipital scalp organ culture, and we cultured till we had anagen and catagen follicles, and we processed these for electron microscopy and immunofluorescence. So what we found out straight away, LC3, one of the key proteins in this process, was highly expressed in the hair follicle. And as we zoom into the matrix, we see particularly high expression, and this very, very punctate expression, which was viewed with confocal microscopy, which is very indicative of autophagy. So this suggests, first off, that the proteins are there in the follicle. So is autophagy actually working? So to try and support this and get a better understanding, we chose to look at electron microscopy, which is one of the best ways of seeing the process is actually occurring. And what we found is these very, very distinctive double membrane organelles, which are actually autophagosomes, and you can see inside the membranes they have probably ribosomes, and what you can see here is the beginning formation of the autophagosome here. As we get to this image here, hopefully you can see there's a fully formed autophagosome, potentially in a fusion process with a lysosome, and here we're getting a degradation process. So taken together, what these images suggest is that we're seeing different stages of autophagy in the human hair follicle, which suggests that autophagy is actually occurring. Then what happens in the hair cycle? Is this just an anagen phenomenon, or is it going on in catagen as well? And what we found is that it does seem to just for anagen here. So if you look at this image here, LC3 expression is particularly high. It's not very great on the presentation. But as we go to early and mid-catagen follicles, this process is shut off immediately. So this suggests to us that hair follicles need autophagy to recycle all of their proteins and organelles during this rapid proliferation we observe in normal hair follicles. So this brought us to our next experimental question. So the proteins are there and the process is active, but does this influence the hair cycle at all? So to look into this, we repeated our usual methodology, but we actually silenced an ATG5, which is one of the key proteins of autophagy, and silencing this inhibits the autophagy process. And then we looked at, again, immunofluorescence, and we looked at the anagen to catagen ratio of these proteins, of these follicles, sorry. So just to show everyone here, so our knockouts are quite successful. This is the ATG5 silencing, and we get a dramatic reduction in ATG5. This is probably about a 60 to 70% reduction in follicles, so we are really stopping this autophagy process. And what actually happens to the hair follicle when we do this, if we look at the matrix of the control, we're getting proliferation, but we compare it to the ATG5 silencing follicles, there is a dramatic decrease, almost cessation of proliferation, and we also get a massive increase in tumor-positive cells. And you can probably see much more clearly here in the graphs that we do get almost a 50% reduction in proliferation and quite a lot of tumor cells. But then how does this affect the hair cycle itself? Well, actually, it causes a huge induction of catagen. So you can see here in the morphological, we've got very strong anagen or early catagen follicles, but in the ATG5, we get much later stages of catagen. And if you look at the graph here, in the knockout controls, we're getting mostly anagen or very early catagen, but when we compare it to the silencing group, there's a huge number of follicles that progress to the mid and late stages of catagen. So together, this suggests that autophagy is active in anagen follicles, and if we stop the follicle from being able to use this process, it can no longer stay in anagen. So it's a very important process in the follicle's normal biology. So that was quite a brief overview of this project. So what hopefully I've shown you today is that anagen hair follicles do use autophagy, and the proteins necessary are in the follicle, and we can view this by immunofluorescence and electron microscopy. And it's particularly important in anagen, and when we go into catagen, this process is no longer used and the follicles turn it off. And if we actually inhibit this process, we get a decrease in proliferation and an increase in apoptosis, and it strongly induces catagen. So what does this actually mean altogether? Well, autophagy is a very important process which we need to look at much more commonly in the human hair follicle, and it's also a new target for cosmeceutical and pharmacological manipulation. And for those of you that have a good memory, our presentation on Wednesday showed that we can actually cosmeceutically manipulate autophagy, and it does prolong anagen. So I'd just like to end there by thanking everyone involved, particularly the Italian Institute of Technology, who have worked really closely with us and were one of our best collaborators for this process, and all of our funding sources involved in this project. Thank you.
Thank you. Questions?
Thank you for your wonderful job. How about the autophagy? If you overexpression it, is that possible to elongate the energy period?
Yeah, that's what we did with the cosmeceutical manipulation. So if we upregulate autophagy, we seem to prolong anagen.
That's good. Thank you.
Can I ask a question?
Yeah.
Catagen involves a lot of tissue destruction. Why would you think autophagy was turned off when there's such a lot of tissue remodeling to be done?
Initially, I thought it would be more involved in catagen, but when we didn't see that and we thought more about it, it's usually for proliferating cells and differentiating cells. And because the follicle in humans is so long-lived, I think it needs to recycle all of these components to be able to keep doing that for such a long period of time. And in catagen, the cells are dying, they don't need the things anymore, so they just get rid of them.
Great work. Does your evidence suggest that induction of autophagy, or turning off autophagy, is that active regulation of initiation of catagen, and as a key regulator?
I would, as a key regulator of catagen
process, yes, and is the DP involved in this, like some form of timing and active signaling or signaling, non-regulation of signaling that then leads to autophagy?
I wouldn't know about the dermal papilla and autophagy. We don't generally see autophagy in the dermal papilla, but it may be secreting something from the dermal papilla. We haven't looked at that. But I do think autophagy is one of the key regulators of anagen, and I think when it's inhibited, catagen's entry, so I think it's one of the main processes involved in catagen entry.
I guess the key question is still how is the timing regulated?
Yeah, that's the key question of all the hair cycle, I think.
Last question.
More comment, please comment that consider several factors involved in catagen, including P53, PIN3, also they are also involved in regulating the autophagy. And moreover, what is more important is that recently it has been shown that keratinocyte growth factor can induce or have a role in controlling autophagy. So I guess that there is a connection. So, well, just a general comment that autophagy is not a biological process per se, is connected with several processes. And during the conference, we had a number of examples that connect. For example, I listened to the proactivity of vorinostat, that is an HDAC inhibitor. One of the major roles of vorinostat is to induce autophagy. We listened to connection with metabolism, MYC, oncogenic MYC, that has a role also in metabolism. It controls autophagy, also P53. So I guess that There will be more to study, but I guess that your point is very good, and I guess there will be a lot of connection with other factors regulating anagen capacitor transcription that they need autophagy in order to do that.
Thank you for your comments, and I'd like to close the session by thanking all our speakers from this morning and remind everybody that now there's the Hair Research Society meetings. Thank you very much.